Hello everyone. In the year 2023, in agriculture optional, they have asked one question, and the question was related to parasitic weeds. The question was classify parasitic weeds on the basis of their parasitism. Also explain the preventive measures and the integrated management practices for Orobanche. It was a 20-mark question and it was asked in the year 2023. So let's see how to solve this question. So first of all, a brief intro about the parasitic weeds. You might have seen the plants like this. So in which it was called as host. In which it was called as the host. And the weed was called as parasitic weeds. Parasitic weeds. So these weeds derive their nutrients from these tree species or plant species. So it may be either totally dependent on the tree species or it may be partially dependent on the particular tree species. So based on this, we can categorize the parasitic weeds into 4 groups. You can use the shortcut like CLOS, close. We will use the term close. So just write down the E term. Here C denotes the Cascata and L denotes the Loranthus and O denotes the Orobanche and S denotes the Striga. So Cascata was the major weed found in alfalfa. And Loranthus was found in mango. Orobanche was the major weed found in tobacco. And Striga was the major weed found in sorghum. So these are the host plant. And in the left hand side, they were called as the parasitic weeds. Parasitic weeds. So it starts from total stem. Cascata was a total stem parasite and Loranthus was a partial stem parasite. Orobanche was a total root parasite and Striga was a partial or hemiroot parasite. Partial or hemiroot parasite. So now let's see about the Orobanche plant. So as I have told you already, Orobanche is the total root parasite. Total root parasite. You can see this plant was called as the weed, parasitic weed, that is Orobanche. And this was the host plant, which was tobacco. Did you find any chlorophyll in the parasitic weed? So it does not have any chlorophyll in it. So it was called as the achlorophyllus. Achlorophyllus. That is devoid of chlorophyll. Those weed species which were achlorophyllus were totally dependent on the host plant. So they depend the host plant for the nutrient and other food material. In India, these parasitic weed were found in the tobacco tracts. Entire tobacco tracts it was found. So they draw their nutrients from tobacco through the establishment of a specialized structure called Astoria. So it was also found in other host species like potato, tomato, and brinjal, sunflower, and safflower. These 3 belongs to Solanaceae, and these 2 belongs to Asteraceae. So I hope it was clear. So regarding the preventive measures, first and foremost thing that we have to follow is use the clean crop seed. Mostly if possible, use the certified seed so that it will be free of or devoid of any weed species. The farm implements such as tractor, combine harvester, and other implements has to be cleaned properly. If a farm implement was used in one field, for example, it was an infested field. Weed infested field. So before moving into the other field, it has to be cleaned properly. The farm implement has to be cleaned properly. And the third step which we have to follow to prevent the entry of parasitic weeds was quarantine. It prevents the entry from one country to other country through international quarantine. And within the country, to prevent the entry from one region to other region, we have to follow the domestic quarantine. So avoid grazing animals. Like cattle and other livestock. It has to be avoided in the infested field. So see what it will do. It will feed on the parasitic weed infested field, and it will transmit the weed to other non-infested field. Non-infested area. So to avoid that, we have to avoid grazing animals in the area where these weed species were found to be more in number. And also we have to avoid the transfer of soil from infested area to a non-infested area. For example, we were doing any pot culture. If you are doing any pot culture, we should avoid the use of soil from these infested area. So there were other wild host or weed species which has to be eliminated in and around the cultivated area, because these wild weeds or other plants can also serve as the host for that parasitic weed. How the Orobanche seeds were germinated in the field, so that we will see, and later we will see how we can use the natural stimulants as a preventive measure. So usually this area I am focusing. This is tobacco plant. And this is the Orobanche. The soil will have a lot of seeds of Orobanche. The soil will have a lot of seed banks of Orobanche. The host plant will release some natural stimulant. Which includes the strigolactone. So these stimulants will trigger the germination of weed species. These stimulant will trigger the germination of Orobanche. This was a thing which was happening in nature. So what we will do using the synthetic stimulant or other stimulants, what we will do here, the seeds in the soil were stimulated using the synthetic stimulants. These synthetic stimulants includes GR7 or GR24 or GR45. So because of these stimulants, the seeds will initially germinate. Like this, but it was not able to find the actual host. So therefore, the seeds will germinate, but at the later stage, the seeds will germinate. But in the later stage, it was not able to find any host. Doesn't find host. So therefore, the seedlings will be killed. This method is called as the suicidal germination. So now integrated management of Orobanche. In this first technique is soil solarization. For soil solarization, we will use the cover of polythene sheets like this. So it will increase the soil temperature. It will increase the soil temperature around 4 to 5 degrees Celsius can be increased. So because of this increase in temperature, the weed species will be killed. The weed species in the soil will be killed. So this was one of the practices which was followed in the cultural method. So other method is hand pulling. So what we will do here, using the labor, we will remove the weeds. So it can be followed for 3 to 4 times per season. And the third technique which was included in the cultural method was crop rotation. Crop rotation with rice. Crop rotation with rice. How it kills the weed species? See, in rice, for growing the rice, we will follow a flooded condition. Flooded condition. So this flooded condition does not allow the oxygen to enter into the soil. So it thereby kills the weed seeds or doesn't allow the weed seeds to germinate. The next method which we can use is trap crops. So mostly the pepper and sunflower were used as the trap crops. So what it will do is, initially it will allow the germination of Orobanche. Initial germination of Orobanche, but later it will be killed. Later it will be killed. We can also use the ground seeds of sunflower for suicidal germination. Suicidal germination we have seen in the preventive measures. We can use the ground seeds of sunflower for suicidal germination. So host plant resistance was the next step or next method we can follow. So in host plant method, we were using either crop breeding tools or biotechnological applications. So using these techniques, we will develop mostly the herbicide tolerant crop variety. Herbicide tolerant crop variety. See, this was the chlorosulfuron resistant tobacco. So the tobacco plant in the field was resistant to chlorosulfuron, whereas the parasitic weeds were susceptible to chlorosulfuron. You just keep one simple relation between nitrogen and weed infestation. When there were low nitrogen content in the soil, weed infestation will be high. Weed infestation will be high. On the other hand, high nitrogen in the soil will result in low weed infestation. Low weed infestation. How this happens in the soil? Actually, high nitrogen will promote the growth of crop. High nitrogen will promote the growth of particular crop. So thereby it can compete easily with the weed species. It can perform better than the weed species under high nitrogen. So the last method, that is biological method and chemical method. So using the biological method, we will use the insects like Phytomyza orobanche. So this was the Phytomyza orobanche at different stage. So these insects will affect the Orobanche weed. These insects will affect the Orobanche weed. Thereby, the weeds will be killed. And we can also use fungus like Fusarium oxysporum. And based on the fungus, they were developing product like product F. So this product F can directly used to control the Orobanche. So certain chemicals were also used to control the Orobanche, but the Orobanche lacks chlorophyll. So thereby using the chemical method was not much easy than like other crop. So soil fumigation can be used using the methyl bromide or methane. Pre-emergence and post-emergence control can be done using the oxyfluorphane. And for post-emergence application, we can use the kerosene oil. And lastly, we can use the allyl alcohol as spray drenching. So this allyl alcohol will kill the tender shoots. Can be applied over the tender shoots. So thereby weeds can be eliminated from the field. So that's it. Thank you for watching the video. Please subscribe the channel and join the Telegram channel. The link was provided in the description. Please join this Telegram channel to get more updates. Thank you.